Hello all, namaste and welcome to lecture 6 of this Inorganic Chemistry Group D, Analytical and Instrumental based on the syllabus of MSc Chemistry, Tribhuvan University. This is Damodar Koirala. So this is the 6th lecture. As I have mentioned in previous lecture, lecture 6 and lecture 5 are the introductory lecture, and here we'll be talking the basics of the techniques that we'll be discussing in future, the lecture 7 through lecture 13. So lecture 6 will be the continuation of the lecture 5, which is also the introduction for rest of the topic. So let's start from where we ended the lecture 5. So this is the slide which shows the significance of atomic absorption spectroscopy and the elemental detection. The elements highlighted on the pink color are those which can be detected using atomic absorption spectroscopy. As you can see, there are several elements that can be detected using atomic absorption spectroscopy. So certainly it has several, the technique has several advantages. So let's understand what atomic absorption is. So when a beam of polychromatic radiation passes through a medium containing gaseous atoms, only a few frequencies are attenuated by absorption. So which when recorded is unique for unique atoms. So here you have the polychromatic radiation, which is the radiation with several wavelengths. Okay, it's a part of the electromagnetic radiation for now. When they are passed through the atoms, gaseous atoms, so these circle are the gaseous atoms, there is the interaction between the atoms and the electromagnetic radiation, which is the spectroscopy. Some of the wavelengths are absorbed by the atoms to move the electron from ground state to the excited state. So those electromagnetic radiation or the electromagnetic radiation with that wavelength, they will get reduced or they will get attenuated. The amount of those electromagnetic radiation will get lower, it decreases, and only some of them will reach. Here, this picture, it shows that the pink color, it is lots in here, it gets attenuated. See, it is less than what we started. Similarly, the blue color, it is less than from what we started. However, the other colors, they were not absorbed. So this diagram, it shows that the blue color and pink color were absorbed by the atoms. So when we measure the electromagnetic radiation that passed through the gaseous atoms, those recordings are unique for unique atoms. That's how we can identify or we can detect the atoms present in that sample. So again, this is how the flow diagram of the instrument. We have the source of electromagnetic radiation. Then there is the compartment where there will be the interaction between the atoms and electromagnetic radiation, and you detect the electromagnetic radiation that comes out of that compartment. Atomic emission. When atoms absorb energy, that they get excited to higher electronic state, these short-lived excited atoms relaxes to the stable ground state by emitting photons with only a few frequencies, which when recorded is unique for unique atoms. So here you have the source of electromagnetic radiation. When when they interact with the gaseous atoms, some of them are absorbed by the atoms, which is the absorption, and the atoms are excited to the higher electronic state. However, these electronic states are very unstable and they relax to the ground state, which is the stable state. While relaxing, they release the energy, the energy difference between those states. So that released energy is detected. Okay, here those released energy is detected, which will give the information about the atoms present in the sample. So here red, green, and blue color are released. Okay, so what happens in this compartment where the interaction between the atoms and the electromagnetic radiation occurs is, first you have bunch of atoms. Okay, when the source of electron energy, which could be any, we'll be discussing about the source of energy in later slides. When they come in contact with the atoms, these atoms, they get excited into higher energy state. Here, they went to this state with, when they absorb red color, they went to this state when they absorb green color, and they went to this state when they absorb blue color. Again, you can see some of them went to red, some of them went to green, and some of them went to blue. However, these are unstable state. Okay, so they come back. They come back. While they are coming back, they release the photon. So that photon, the energy difference is what they observed earlier. So they release the red light, red color, green color light, and blue color light. And these light will be detected. Once detected, you can determine the atoms present in the sample. Okay. So this slide shows the comparison between atomic absorption and atomic emission technique. So what happens is, in both cases, you have the source of energy. Okay. And in both cases, there is the interaction between the source of the energy and the atoms. Okay. However, the detection is the difference. Here you detect the attenuated electromagnetic radiation. Okay, what the wavelength of electromagnetic radiation that were absorbed. That's what you detect here. Here you detect the electromagnetic radiation that are emitted. Okay, here you measure what is absorbed, what is attenuated, what get reduced. Here you measure what is emitted. So again, in atomic absorption spectroscopy, it is the one-step technique where you give bunch of you give the electromagnetic radiation with bunch of wavelength. Then some of them are attenuated and you measure it. However, in emission, it is the two-step technique. Here, first the atom are excited. Okay, that is not measured though. What is measured? When those excited atoms come back to ground state, that is measured. That is two steps: excitation and then emission. Okay. Again, the difference is attenuated EMR is detected and emitted EMR is detected. So here is the case of the sodium. So in sodium, the ground state electron, the valence electron is present in 3s orbital, which when absorbed right energy, it can go to 3p, 4p, or 5p. Okay, so here it requires, as you can see, it requires 590 nanometer to go from 3s to 3p, 330 nanometer to go from 3s to 4p, and 285 nanometer to go from 3s to 5p. So when it absorbs that is absorption technique. If you measure what wavelengths are absorbed. However, when it is in the excited state, it can also come back to ground state by emitting the same amount of energy, and it is emission. This is the type of spectra that will observe in absorption. Okay, you can see this is one of the wavelength that is absorbed, this is one of the wavelength that is absorbed, and this is the one of the absorbed wavelength that is absorbed. So this diagram, it shows that most of the absorption occur in 590 nanometer range, then the 330, and then 295. The height we'll be discussing in detail in later lectures. However, for now, you can say that the height will also give you some information about how much. Okay. So this is the absorption spectrum for sodium vapor, sodium atom. However, you should also note that the wavelength of absorption and emission are identical for atoms. So in case of atoms, whatever the wavelength it is absorbed, the same wavelength will be emitted as well. Okay, so you might have question then why we have to do absorption or emission, why cannot we just do one of them? There are both advantages. There are several advantages on both techniques, which we'll be discussing in detail in lecture 7, 8, and keep forward, okay? So the absorption and emission spectra for sodium are relatively simple and consist of only a few lines. Okay, these are the lines. As you can see, there are only few lines. For elements that have several outer electrons that can be excited, the excited absorption and emission spectra may be much more complex. So what this is saying is in case of sodium, there is only one valence electron. So the lines are very simple. However, if we are doing other elements with multiple valence electron, then the spectra will be little bit complex. Okay. However, now for example, if you want to detect if you have sodium in your sample or not, then you do the you obtain the atomic absorption spectrum or atomic emission spectrum, and you compare with the standard sodium. Then you compare the spectrum. If the spectrum for the sample is also in similar height ratio at same wavelength, then it will confirm that your sample has sodium in it. Otherwise, it will confirm that your sample does not have sodium in it. Okay, that is qualitative, qualitative analysis. You can also do quantitative analysis. How much sodium is there in your sample, which we'll be discussing in future slides. So that gives you about the technique that is absorption and emission. What is going on in absorption and what is going on in emission. Now we are focusing toward the instrument part. The instrument is called optical instruments. So they are called optical instrument because there are several optics that are used, optical parts like lenses and glass that we talk in optics lecture. They are used in this instrument. That's why it is called optical instrument. Those lenses and glass, those optical components are used to guide the electromagnetic radiation. Okay. So the instrument is the communication device between the system under study and the investigator. So we are the investigator. We human beings are the investigator, and the instrument is it's a communication device between the system. The system is our sample. Okay, that we want to study, that we want to analyze. It is the instrument act as a communication device. So what happens in the instrument? There is a source of energy, like I have shown in earlier slide for absorption and emission. There is the source of energy, and then you have this system under study. So the system under study in our case is atoms. The atoms are in gaseous states. Okay, you have atoms, and then you have a detector to detect the electromagnetic radiation. That will be considered as a response. The response we get. Okay, this is how the instrument flows. So first you have non-electrical information. So the domain is the information. So initially you have non-electrical information, okay, that we have. And then that the instrument converts into electrical information. However, when it gives us the information is again non-electrical. Okay, for example, we have a sample which is non-electrical, right? We put that sample in the system. Then whenever the instrument converts the information in the sample into electrical information, the electrical information like voltage, current, or charge, it measures those information. And again, when we get the information, we do not get what voltage or what current or what charge. We get how many gram per liter, how many, what mole per liter. is there in the sample. Those are again non-electrical information. Okay, again non-electrical information, the instrument converts into electrical information, and those electrical information again they are converted into non-electrical information for us. That's how it acts as a communication device. So there are 5 major components in all optical device. So for all the optical instrument that we'll be discussing now on, whenever it is asked, you should be familiar with these 5 components. These are very common components, and these are the components that we'll be discussing today. And in near future, whenever we talk about the specific instrument. So today we'll try to understand what these components are. And in future, whenever we talk about the instrument, we'll go in pinpoint to say what kind of source is used, what kind of sample holder, what kind of wavelength selector is used, or what is the difference between 2 instruments, what are the difference in the component, those things, okay. Now source is a stable source of radiation energy at the desired wavelength range. So basically source is the one that provides you the electromagnetic radiation or other forms of energy, okay, any forms of energy that will be in the desired wavelength. Okay, sample holder. So sample holder is a transparent container used to hold the sample. Okay, so basically they are used to hold the sample. In our case, since we are studying the atoms, the sample holder is usually the free space inside the instrument, okay, where the atoms are made. Wavelength selector. So wavelength selector is a device that isolates a restricted region of the electromagnetic radiation spectrum for measurement. So basically, like it's a selector, means that device, it will select certain wavelength or certain range of the wavelength. That is called wavelength selector. Photoelectric transducer. So in previous slide, I showed you there are non-electrical information and electrical information, right? So this is the device that converts the electrical information into non-electrical information and non-electrical information into electrical information. Transducer. It is also the detector. We call it detector. Okay. Signal processor and readout device. So the information that we get are usually in very small amount, which will be very difficult to read or to analyze, to detect. So those information are usually amplified, and that the amplification is done by the signal processor. And we get the information in computer screen or in certain other form of screen, okay, that is called readout device, where we see the information, which in most of the cases the computer screen monitor. Okay, in coming slide we'll be discussing these 4 topics in detail. However, the 5th one, which is the signal processing and readout device, it is not discussed. Okay, it is just the screen. So those 5 components can be arranged in several ways. Here are 3 ways it is shown they can be arranged. Okay, so the first one is you have source. From that source you get the wavelength of several ranges. Then you put the wavelength selector where you select only certain wavelength of energy. Those certain wavelength of energy goes into the sample where the interaction occurs, and then depending on what we are studying, that will be detected. Okay, what kind of wavelength is observed or what kind of wavelength is emitted. Once detected, we get the information from the signal processing and readout device. So that's how it is arranged. This is one of the way that the components can be arranged. The second way is, see, it is linear here. The second way is you have source, Then you have wavelength selector. Then there is a sample. However, the other component is again wavelength selector, which is perpendicular to the previous wavelength selector. Okay, whatever information we get from the sample, it travels in 90° to the previous component. And detector and readout device. So again, what's going on here? You select certain wavelength at the beginning that will interact with the sample. Then you have wavelength selectors that will select certain wavelength only that you want to detect. Here you don't want to detect everything. You only want to detect certain wavelength. How do you select those certain wavelength? By using wavelength selector. Okay, and the detector will detect how much or if or not there is that wavelength present in the wavelength, the electromagnetic radiation that came out of the sample. This is the second way you can organize your components in optical instrument. So the third way is you have the sample in a container. Okay. Then the source is also within there, same. The source and sample are in the same container. Then there is the interaction already occurring. After there is an interaction, certain wavelength will be emitted. They are filtered or they are selected. The wavelength selector selects the desired wavelength and send those wavelength to the detector, and detector will detect them. Once they are detected, they are sent into the signal processor for amplification and for readout information. Okay, in this case, source and sample are in same compartment. They are in same compartment. Okay, now in future, whenever we discuss the optical instrument, the specific optical instrument, you should also able to recall these diagrams and see in which category that does that fall. Okay, where that fall. So in general, this is the setup for absorption instrument, and this is the setup for emission instrument. Okay, so here you have the source. That source of goes into the sample, the source of energy. The energy goes into the sample. Then there is the interaction. Out of this interaction, you select only certain wavelength. Those certain wavelength goes into the detector. So the photoelectric transducer is the detector, and from that detector, you convert those information into electric signal, and it goes into the signal processor and readout device. So again, you have H mu, which is the energy coming out of the source, that goes into the sample. You have H mu coming out, goes into the wavelength selector, H mu coming out. So these all H mu are the non-electrical information, goes into the photoelectric transducer, converts that into the electrical information. I is the current. So this converts the non-electrical information into electrical information. Now that electrical information is analyzed, amplified and analyzed by signal processor, and we get the information that is non-electrical from readout device. Okay, this is the general setup. For emission, so this is general for absorption, and for general setup for emission is the third one that we saw in previous slide, that you have source and sample holder in the same compartment. Okay, the source is the source of the energy. Then you get the information that is H mu, goes into the wavelength selector. That there you select the H mu, which H mu you want to detect, goes into the photoelectric transducer, get converted into the electric information, which is current, and the signal processor and amplifies the current and detect and give us the information in non-electrical information format. Okay, so these are the general design. Now let's get into the detail on those 4 components. Okay, one of the last one is ignored. because it is just the computer monitor or the digital readout device. So the source of the energy. So what are the different sources of the energy that we can be using, okay? So one of the sources is electromagnetic radiation. So it must provide an output that is both intense and stable, okay, very high, but stable in the desired region of electromagnetic spectrum. So maybe we just want UV, then it must provide the electromagnetic radiation in UV region, which is both intense and stable. So it can be classified into two forms. One of them is the continuum source, and the other one is the line source. So the continuum source is the radiation over a wide range of wavelength, maybe 200 to 300 nanometer, for example, 200 to 1000 nanometer, 500 to 600 nanometer. That is continuum source. Okay, you need all the wavelength in that region. The other one is line source. So the line source is, it emits a radiation as a few selected narrow wavelength range, very small wavelength range. That will be line source, like the line where you have very particular line, right? So for example, hollow cathode lamp that we'll be discussing in detail when we talk about the atomic absorption, it will provide you single wavelength, for example, or 237, 530, and 647, those wavelength, okay, line. So these are some of the example of the sources in electromagnetic radiation. So you have hydrogen and deuterium lamp. They provide continuous source in the range of 160 to 380 nanometer, and they can be used for UV molecular absorption. You have tungsten lamp, which is also the continuous source. In this range, we can be used in the visible molecular spectrum. All sorts of lamp, continuous for IR, IR. And there is this hollow cathode lamp, which is for atomic absorption. So remember, we are studying the atomic spectroscopy. So this last one will be very helpful. We'll be discussing about the hollow cathode lamp in detail when we talk about the atomic absorption. Okay. So other sources of energy are thermal energy. Thermal means heat, right? So of the thermal energy, there are two common sources, that is flame and plasma. Okay, flame, it is obtained by the combustion of fuel and oxidant, such as acetylene and air, to achieve the temperature of 2000 to 3400 Kelvin. So the flame is same as that we have in our gas stove, in our kitchen, or in our lab, chemistry lab. That is the flame source. The plasma are the hot ionized gas which provide the temperature of 6000 to 10,000 Kelvin. Okay, it is very hot ionized gas. As you can see, the flame, the temperature range is very low, which is only up to 3400. However, the plasma are up to 10,000 Kelvin. So when we discuss about the emission spectroscopy, atomic emission spectroscopy, we'll be discussing in detail about the flame and plasma. The flame we might also discuss in the atomic absorption spectroscopy. However, the plasma we'll be discussing in atomic emission spectroscopy. These two are very powerful source. They're very useful in emission technique. Okay. Chemical source of energy. So the energy that is obtained from exothermic reaction. They are used in chemiluminescence, where the analyte is raised to higher energy state by means of chemical reaction, and emitting characteristic radiation when it returns to the low energy state. So this is like the emission spectroscopy, where the reaction occurs, which is exothermic reaction. So the energy is released by the reaction, and the molecules in there, they absorb those energy, goes to the higher excited state, and when they come relaxed to the lower energy state, they emit certain electromagnetic radiation, which will be detected. Okay. When the chemical reaction results from a biological or enzymatic reaction, the emission of radiation is called bioluminescence. Okay, if the energy is by biological or enzymatic reaction. So these are the sources of energy. One of them is the electromagnetic source for electromagnetic radiation. The other one is for thermal energy and the chemical source. Now, the wavelength selector. Okay. So the wavelength selector outputs a limited, narrow, continuous group of wavelength called a band. So basically, the purpose of the wavelength selector is you might have where the electromagnetic radiation or the energy of very larger range, for example, 10 nanometer to 5,000 nanometer of wavelengths. However, you only need the wavelength from 200 to 400. So you don't need all the wavelengths, right? So what you do is you select those certain wavelength using wavelength selector, and you pass it into the sample for the interaction. So that is the purpose of wavelength selector. Also, say you already know what wavelength are to be emitted by certain atoms. So what you can do is, for example, if you are detecting sodium, then you can only specifically select the wavelength that corresponds to this emission of the sodium and detect them. So you have desired information only. You don't have to worry about all the wavelength. So how do you do that? You use the wavelength selector. Okay. So of the different types of wavelength selector, the first one in here is the filter, like the filter that we use for filtering the water. Okay. So there are two types of filter. One of them is the interference filter. So the interference filter, it transmit or reflect ranges of wavelength. Okay, so it transmit the desired wavelength and it reflect the undesired wavelength. That is called interference filter. Absorption filter. So what does the absorption filter do? It blocks. Here it reflects, however, it blocks by absorbing those wavelengths that are not wanted. So it transmits the required wavelength, however, it absorbs the unwanted wavelength. Here it reflects the unwanted wavelength, here it absorbs the unwanted wavelength. So this figure shows the comparison between interference filter and absorption filter. As you can see, if you are using the interference filter, then the bandwidth, which is the width, half height width. Okay, so in half of the height, you take the width of the curve, that is called bandwidth. So the smaller the bandwidth, better is the filter. Here the bandwidth is 10 nanometer. For example, if you desire the wavelength of 448 nanometer, then you get the wavelength from 440 to 460. Okay. Yes, that's what you get. Absorption filter, however, if you want 500 nanometer, then you can see the bandwidth is 50 nanometer. In the half of this curve, half height of the curve, the width of the curve is 15 nanometer, which is larger than the interference filter. So if you compare these two, interference filter is better than the absorption filter, which also means that interference filter is more expensive than absorption filter. So depending on what is your desire on the chemical analysis, you have to choose which types of filter you want to use. Okay, like I have discussed on selecting the analytical method, here also you have to select which filters you want to use. So another types of filter is monochromator, and this monochromator is what we'll be hearing a lot in these lectures. So the monochromator is the wavelength selector that can continuously scan a broad range of wavelength. Okay, it continuously scans. That means it continuously changes the wavelength. It can, okay. Used in most scanning spectrometer, including UV, visible, and IR instruments. So scanning instrument where you want to scan all the wavelength, that's where the monochromator is used. So let's look into it, what it is, okay. First, the mono, in general, the mono means single, right? Chromator means wavelength. So the purpose of monochromator in general is to isolate the single wavelength. So 498, you will get 497.5 to 498.5. 1 nanometer wavelength of the electromagnetic radiation. But how it is observed? Say you have a sunlight, which is a white light. When it strike the diffraction grating, which is like a mirror at certain angle, okay, then that sunlight is separated into several colors, right? Violet, indigo blue, green, yellow, orange, red. However, once they are separated into the different color, then this is where our sample is located, or the detector is located, okay, which we'll be seeing, like I showed you during how the components are arranged. On this side, you will have, for example, the sample. Now you want to interact the sample with green light. Then what is going on? Here, this diffraction grating, you can rotate it at certain angle. So you rotate in such a way that these light will pass, or the wavelength of the light will pass simultaneously, one at a time. So now the green is passing, okay. So the interaction between the sample and the green color occurs. Then after that, next green will transfer, next green, then comes the time of yellow. There are different yellow colors, then red, different colors, right? That's how it will help you to transmit only one color. All others are blocked. As you can see, there is a small hole through which a narrow wavelength of light can be passed. This is the monochromator. Okay. It can continuously scan because as you rotate this slide continuously, okay, you have a motor which will rotate this diffraction grating at certain angle, and the rate of the change of the angle will be very small. And once it is rotating, only one wavelength of the light passes here, then it rotates automatically, then the other wavelength, then the other wavelength. So it is continuously changing. It is continuously scanning. Okay. Now we get into the detector. So earlier, your human eyes were used as a detector. But in current devices, modern instrument, they contain a device that converts radiation to the electrical signal. So you have radiation, which is electromagnetic radiation. Though that radiation is converted to electrical signal, and this electrical signal is measured. Like I showed in previous slides, the radiation was converted into current, and that current is measured. Okay, let's see how it is done. So one of the types of the detector is photon detectors. So the photon detectors is the detector that can detect photon. Okay, it can detect the photon. So there are several types of photon detectors: vacuum photon tube, phototube, photovoltaic cell, photomultiplier tube, silicon photodiode. What we hear in commonly is photomultiplier tube, PMT, photomultiplier tube. So here is the example of the working principle of the photomultiplier tube. Say you have certain photon, okay, and you know the wavelength of this photon, or even you don't know, the instrument will know what wavelength of the photon is this. Now we want to measure the intensity of this photon. So what happens is this photon hits the photocathode. So when it comes in contact with the photocathode, the electron is released from the cathode, okay, due to photoelectric effect. Okay, when the photon hits the matter, electron is released due to photoelectric effect. Then those electrons are attracted to a dynode. Okay, see, the electron is attracted toward the dynode. Here the electron comes in contact with the dynode and more electrons are released. Okay, then those again travel into the another dynode and even more are released. As it keep traveling, they keep interacting with the different dynodes. The number of electrons keep increasing. It is called cascading of electrons. Okay, it is increasing exponentially. By the end, when it reaches the final anode, you can see one electron, which was very difficult to be measured. You cannot get much information from one electron. It is converted into millions of electrons. Okay, one electron is amplified into millions of electrons. Now you have millions of electrons. You can measure the current and you can detect what is the intensity of the photon. Okay, this is proportional, right? For example, if you have 10 photons, then you have large number of electrons coming out in the first place, which will go to the first dynode. It will release larger amount. By the time it goes, it is proportional. Okay, that's how the photo multiplier tubes works. Starts with very small amount of photon, turns into the detectable amount of the electric information. So that was the photon detector. This one is the thermal detector. It is usually used for IR spectroscopy. Okay, so here thermal means heat. So it is used to measure the heat. We'll be using these types of detector very little. So there are the three types is thermocouple, bolometer, and pyroelectric transducer. So just understand that this is temperature dependent, okay, and detects the heat. So these are the several detectors and their characteristics. There are mainly photon detector and thermal detector. This is the wavelength range that they can work. Okay, so you should select which detector you want to use for what purpose. And the output signal is current or change in resistance or voltage, could be voltage or membrane displacement or current. So these are still the electrical information. However, when we user get the information, those are non-electrical information. Okay. However, what you need to understand is the initial information is photon, which is non-electrical. It is converted into current, which is non-electrical, and then it will also be converted into non-electrical information. Okay. Sample holder. So in general, the sample holders are the containers where we put our sample. They are called cells or cuvettes. You might have seen these types of devices in your bachelor level also. Okay, there are a few types: quartz or fused silica, silicate glass, crystalline sodium chloride. So depending on what these containers are made of, they are classified into their types. Okay, so quartz, it is used for UV and maybe for visible region. Silicate glass, they are cheap, okay, and they can be used in UV. And crystalline sodium chloride, it is used for IR. Okay, so these are the four components of the instrument. So this is the summary of what we have discussed. okay, the source sample and sample holder detector and various electromagnetic region. So these are the region. You have ultraviolet, visible, and infrared. If you want to do the ultraviolet spectroscopy, then you need to have a source, which is deuterium lamp. For visible, tungsten lamp, and for infrared, North Star. And accordingly, you need to have this sample holder, and you can have these detectors. Okay, with this, I would like to end this lecture here. So now we got the introductory part of what we'll be discussing in future. These topics I just want you to be familiar with, get used to with the terminologies. So when we discuss the main content, there is no confusion. Okay. With that, I'd like to thank you for your time and see you in next lecture.